we're looking here, we can see that the cells are not perfect plant rectangular shaped cells that you think of. Like a lot of folks will, if you look up a classic plant cell online, often you see a square or rectangular shaped cell. These onion cells do almost have that look and almost seem like bricks in a brick wall kind of thing. We can kind of say that, except not perfectly rectangular. Kind of funky, a little bit funky shaped on the sides. And I had you stain them with iodine like you can see here, and you can really make out that cell wall and nucleus. Some of the space in here is just vacuole, and other components that are kind of tricky to make sense of. There are plastics in there, though we can't really see them, and they're translucent. If they were chloroplasts, they'd be what color?
Green.
Yeah, green. And because you added iodine, if there were amyloplasts, they would be what color?
Purple.
Like a purpley black, because what do the amyloplasts do?
Absorb starch.
Yeah, they create and store starch. So, and sometimes on these nuclei, you can kind of see these darkened blobs in the nuclei. Do you remember what those are called?
Nucleoli.
Nucleolus. And major function? Ribosome production. Major function of the cell wall?
Protective.
Structure, protection, rigidity of the cell, even if the cell membrane, something comes with that cell wall, depending on how thick that cell wall is, it will stay there. So we saw when we looked at this image here, we can see that cell wall can sometimes be very thick. And actually a primary layer, cell wall layer, and then a secondary cell wall layer, and it's really going to help with that, provide structure to where you've got material like that hard, bony, a coconut, right? Coconut shell, and a peach pit. We're talking like really hard. All right. You guys didn't see this image, but just to review, if we were to kind of take this cell here, here's you guys remember what this light colored line is here?
It's like.
You guys remember this one?
I should have my lab out.
It's kind of this material that sort of sticks the cells together. Middle lamella, very good. All right. Do you guys have a question? No? Okay. Let's keep the chatter down so we can all stay focused better. All right. So we got middle lamella. If we were to kind of cut this open and pull that cell out and look at it kind of head on, we would see something like this, where we're seeing the cell wall, which would be all this kind of lighter colored material, and then these little eyeball looking things are the tunnels connecting cells. What are those called? Plasmodesmata. So I'm just doing a quick review here. Make sure guys, we're all on the same page here and kind of remember some stuff from last time before we jump into more plant cell stuff and plant tissues. All right, if we then, instead of opening this up, looking head on at the cell wall, instead we do a cut right through here and look, this is the kind of thing we would potentially see, something like this, where you've got a fine line, middle lamella, this light colored area, this is one cell wall, one cell, another cell wall of the next cell, and then the tunnels through the plasmodesmata. And then the inside of cell A, inside of cell B, kind of thing. Those images make sense to you guys? You guys good on that? All right. Cool. So you looked at the leaf of Elodea, And in water, you're really looking at those chloroplasts, cell walls, this empty space here, vacuole. Look, what do you guys think that is? Can you guys see my pointer? It's not very good. Right there. Right here,
That's
this little blob, this like, it is, so the vacuole is all this clear space here. That's all vacuole, and you can tell because the chloroplasts are kind of like around these spaces. And that is actually vacuole. Right here, that is nucleus. And then when you added the salt water, the cell membrane, you can kind of make sense of the cell membrane at that point and see that the chloroplasts are kind of tight together. Cell membrane is right around all those chloroplasts. It's like a balloon that lost a water balloon that lost all this water from inside. It's just kind of tight around all the organelles. And the vacuole loses its water too and is in there as well. So looking at the potato, here we see, hopefully you guys could see what's this line right here? Let me use my pointer so you can see better, sorry. All right, here, this line. What is that?
Cell wall.
This fine line right here, cell wall, very good. And there is some kind of empty space there. What would that be? I mean, some of it's going to be vacuole, some of it's going to be the cytosol. And then these here, they're clear, but as soon as you add that iodine is normally a yellowy orange color, but if there's starch present, it changes to black or purple. So we can see that those structures are what?
Amyloplasts.
Amyloplasts. So don't get mixed up and think this is a cell. This is an organelle inside of the cell. And here's the cell, and it is packed full of amyloplasts. This is a special kind of cell called a parenchyma cell, which you're going to hear, which we're going to be learning about today. It's one of the more common kinds of cells you'll find in plants. And it has a lot of important functions, and it's kind of like the background support cell, ground tissue. So this is another type of parenchyma cell that makes up a lot of the material in fruits like peaches, pears, and apples, peppers. They're parenchyma cells. These parenchyma cells have a special function in that they're full. They at one time were full of chloroplasts, and then those chloroplasts converted and changed into what?
Chromoplasts.
Chromoplasts. Very good. Plant crystals. I know that folks had a little bit of rough time finding. I was able to get a hold of a better banana this time, a green, really green banana. So we did see crystals, but you really had to slice as thin as possible on the outer peel and to see them. With a green banana, you can use the very thin outer peel layer, but you can also get even in a little deeper and you can see crystals really well. So I've got that out for you to finish that part up. If you didn't have very good luck, you just came up here and you saw it. I'd love for you to have the chance to like make your own slide and see that yourself. If you see them inside the cell still, this is what it'll look like. It'll look kind of like a dark rectangle, shadow of a rectangle. And that's that vacuole packed full of crystals. If you get to like an edge area where there's a lot of water on the edge of your section, the cell might be broken open, and then you'd see the crystals all kind of spewing out. And it's really cool to see it like that. And sometimes for fun, you can like eat a green banana peel and see if you start to feel like some tenderness on your mouth from all these crystals. Like, it's not gonna really hurt you. You just might feel like, oh, it feels kind of weird. Yes?
What were the crystals for again?
What do you guys think? What do you guys think these crystals are helping to do? If you see them a bit more with a green banana versus, and they start to kind of fade out a little bit more as the banana becomes riper.
Does it like ward off predators or something before it's ripe?
Yes, if, especially like, think of like a smaller organism than us trying to take bites out of that. Now those are sharp. And so, yeah, that's what the thought is to deter eating the fruit when it's not ready to be eaten. If a fruit gets eaten before the seeds are mature enough, then kind of defeats the purpose of seed dispersal, of attracting seed dispersers with the fruit if they're eating it too early and the seeds aren't ready. It also might seem like there are some plants out there that entice seed dispersers to come and eat, and they actually eat the seeds. That might seem like it's not making sense. But in doing so, what do you think happens a lot with that kind of situation? Yeah, some seeds get eaten and a lot of seeds get like dropped along the way. And thinking about, can you guys think of any good example of that where the plant actually entices seed dispersers to eat the seeds? Yes.
There was one in the life video that was covered in this like purple like juice that enticed birds to come in, eat the whole thing, they'd fly off, and potentially miles away they'd lay their droppings.
Yeah, that's a great example where the plant is enticing the seed disperser, the animal, to eat the whole seed, and then it's just gonna defecate somewhere, and then that seed has all this great fertilizer around it somewhere else far away from the parent plant. So then the parent and the offspring don't have to fight over those same resources. Gets it further away. But what if an organism is actually chewing up the seeds? Can you think of any examples like that?
Oak trees with acorns and squirrels.
So what's going on with oak trees?
They make acorns and then squirrels eat the acorns, but they also bury some.
Yeah.
And then they forget about them and then they're just planted.
Yeah, that's a awesome example. So you guys just shared really great examples of different kinds of ways of going about it, either enticing the seed disperser to eat the fruit, including the seeds, but then not actually chew up the seeds, but then the seeds pass through a digestive tract whole and are unharmed, or a situation where the seed disperser is actually eating and destroying the seeds, the baby plant inside the seeds, with the example of acorns. But a lot of those seeds either get dropped or buried somewhere, or put somewhere and then forgotten about. And hence why we have so many oak trees around us, that we live in a oak woodland area. So, right, hand was raised.
I was just gonna say figs are a good example, because they have really small seeds and things that say that animals can eat seeds without branching.
Which one?
Figs.
Oh, right. Yeah, that's a good example. Those fig seeds will pass right through the digestive tract, and yeah, that's a really good example. Okay, so for the tilia, we see some crystals kind of more to the bark, out near the bark area. And what kinds of crystals are these ones?
Right. No, wait, hold on. No, it's the druses.
These ones are the druses, and then the banana have the raphites.
That makes sense.
Catch up later. All right. Okay. So that's just a quick review over that. Let's jump now into what we're gonna jump into today. You are going to be getting a large lab package. Just give you a heads up. Don't freak out. We're covering half of it, and then the other half we'll roll over to Tuesday, when we'll also be jumping into. going over, we're gonna at some point fit in mitosis and talk about that. That you can see that's on the schedule. But I'm gonna probably roll right into continuing with plant organs and tissues, and then we'll be jumping right into root, which is the first plant organ we're gonna be talking about. So plant organs and tissues. Higher plants have 3 to 4 major organs, depending on the type of plant. And when I say higher plants, I don't mean like more important or better plants. I mean, if we're talking about an evolutionary tree, I'm talking plants that have evolved more recently. They're going to be higher up on that evolutionary tree. So, can you think of any plants that would not be considered higher plants? Yes.
Phytoplankton?
Well, phytoplankton, that's awesome because I know, depending on what you're listening to or what you're reading, you will hear people say phytoplankton are plants. They're not plants. But some people will call algae plants. They're not. But phytoplankton, a lot of what's mixed in with phytoplankton are dinoflagellates, diatoms, cyanobacteria, things that aren't in the plant group. But yeah, they would definitely not be considered. They're not plants, but then also they're unicellular too. So so you're right in that they would not have multicellular things like tissues and organs. So that's good. That's good. What else? What else? What's that would actually be a plant that wouldn't have organs? Do you think?
Fern.
Ferns. You're getting there. You're getting closer.
What about like
Keep going in that direction.
Grass and
Mosses?
So weeds, grasses,
Like an R?
ferns.
Weeds?
Actually, those all. Wait. What else you said?
Weeds?
Weeds.
Reeds.
Oh, reeds.
Reeds, yeah.
Shoot.
You can like pull them apart.
Yeah. Okay.
They seem pretty primitive.
They do. Yeah, not primitive. That's a good word. So, more recently evolved would be higher kinds of plants, less maybe that thought of as being less primitive. So ferns, though, ferns have been around for a while, and we see them in the fossil record for a long time ago. So we're talking like 300 million years back. And even further. So ferns, though, do actually have plant organs. They have true stem and true leaves. They have roots. They have organs. And that's because they have vascular tissue. So we're going to be getting into all the major groupings of plants. So think of something that's maybe more primitive than a fern even. Yes. In the back.
Seaweed?
So seaweed, that again is in the algal group. They photosynthesize, but they are not considered plants. But yeah, so they would not, you're right, they wouldn't have organs either. Yes?
Moss.
Moss. Yes. There we go. So moss. Yay! You guys all I love it. I love everybody's comments. It's great. So moss are earlier evolving than even ferns. And they are, they're amazing. They're multicellular. They're plants. They're really cool. Liverworts, mosses, and hornworts. These are bryophytes. We'll be getting into them. Those plants have plant tissues. Of course, they're made up of plant cells, but they don't have their components are not organized into organs. And that is really linked and tied to vascular tissue. So vascular tissue, when you think of like a leaf, and you look at it with the light behind, you see those veins. That's vascular tissue. Those are veins. Mosses, liverworts, hornworts, the bryophytes, they do not have veins. They don't have vascular tissue. But ferns do, right? With a fern frond, a leaf of a fern, there is vascular tissue. So, that you're going to see as we get into plant organs, a root, stem, leaf, a flower, fruit, those kinds of things. you're gonna see that vascular tissue, how that vascular tissue is arranged is different between leaves, roots, stems, and so it's really based on that vascular tissue. So when I say plant-oriented tissues, higher plants have 3 to 4 major organs. It includes everything other than the mosses and the liverworts. That's what we're going to be getting into when we get into organs. Now, mosses do have the cells that we're going to be talking about now, but just alluding to something that we're going to be getting into in more detail later. Yes?
What's the word under the picture? Like after if?
Oh, shoot. I think it says angiosperms. It should say angiosperms. Flowers and fruits if angiosperm. Angiosperms are flowering plants.
How is it spelled?
I messed up with that one. Because I did modify this a little. It looks like I changed that up a bit. All right, so moving on. 3 major tissue systems. Talking about plants and their tissues. Plant tissue systems, okay? And I do kind of a mix of cursive and printing. Do you guys hear that in the state of California, there was some discussion about whether or not cursive was going to be something that was going to be taught in schools? How many of you learned cursive in school?
I was supposed to.
How many of you didn't? That's what I should ask. So yeah, you didn't learn cursive in school. So have you just had to learn it on your own?
Yeah, I taught myself what it is.
Yeah, it's not that hard. I mean, it's a little tricky, but I think it's something that was passed fairly recently that schools have to teach cursive, and it's staying. And I do kind of a mix when I run on the board. So I thought about that. I was like, I wonder if you all just have to get used to how your different professors, how teachers write on the board. It's always different, right? So, okay, tissue systems. We have, first off, the ground tissue system. And we have the vascular tissue system and the dermal tissue system. And actually, I'm gonna write it down, sorry. It's okay, I'm just reorganizing how I wanna talk about it a bit. Dermal tissue. Ground tissue. And vascular. So any plant organ you look at of a cross-section, like a cross-section of a root, you should be able to see all 3 of those tissue systems and make sense of it. And, oh, I'm sorry, I'm just not noticing you. There are extra chairs are pulled in. Let me get one. I apologize, I didn't realize. Come on over. I think that, looks like you guys, there's 4 folks. These tables can definitely hold 5, so go ahead and maybe move it just a bit to squeeze in another person in there. Thank you. All right, so here we have cross-section of a leaf, cross-section of a stem, cross-section of a root. And when we look at each of those, we see that the cells are stained a bit, so maybe you might notice that right off the bat. And so a little bit of staining there, but I don't want you to memorize color exactly. I want you to think about region and where these cells are. And as you remember, I've mentioned when you're in lab and you're doing your drawings, I want you to add color. And it's not so much exactly having to be the color that you're seeing, but color them in a way so that it'll help you remember what it looks like, what the cell looks like, where the cell's positioned. And because you'll be labeling different parts of the cells, different parts of the organs you're looking at and whatnot, okay? So here we have a cross section right through a leaf. This is the upper surface of the leaf, the lower surface of the leaf, and all this stuff in the middle. All that filler is ground tissue. And embedded in the ground tissue are a couple veins, which are kind of hard to see in this section. The veins make up the vascular tissue system. And then it's surrounded by a protective layer of cells, sometimes even multiple layers of cells, called the dermal tissue system. And that's a leaf. Looking at a stem, we've got the same thing. We've got the dermal tissue is the skin of that stem, the epidermis. And then inside of that, you'll have a mix of some ground tissue and then vascular tissue embedded in that. Here we have a root, same thing, dermal tissue on the outside, ground tissue inside with a big cylinder of vascular tissue in the middle. So that one kind of looks like, a little bit like, more like a donut kind of shape, with the middle being vascular tissue. So now, you've got those three tissue systems that you'll see in a plant. Within these tissue systems, there are specific tissue, names for the tissues. So first off, what is a tissue? Tissue is, go ahead, what was that?
A collection of cells.
A collection of cells.
That's
Do they
it.
have to be the same kind of cells?
They serve a similar purpose.
Awesome. Collection of cells. So tissue, collection of cells serves similar purpose. And they can be one kind of cell. They can be more of like a complex tissue where it's multiple types of cells working together for a similar purpose. Okay? So with the dermal tissue system, you have two types. You have the epidermis. Let me make my R a little more obvious there. And periderm. So, I'll go ahead and write these out, and then we will get into each of them in a little more detail. With ground tissue, we've got three major tissue types that are made up of those specific cells that actually have similar names, or the same name, really. And I want you guys to get familiar with how to say them, how to spell them, okay? So the first one is parenchyma. Can everybody say that? Parenchyma. Parenchyma. The next one, cholenchyma. Cholenchyma. And the next one, sclerenchyma. Sclerenchyma. So we've got parenchyma, and then cholenchyma, and sclerenchyma.
Fairly easy to
Okay.
spell.
So we're going to get into what each of those are. This is big picture first. Vascular tissue, you've got xylem and phloem. So again, this is the same thing as I kind of showed before, but before this one, this is showing an actual microscope slides. Here, similar kinds of things. Cross section through leaf, through stem, through root, but diagram. So it's a little easier to make sense of things when it's drawn out like that. You guys agree? Comparing that to that. All right, so for a leaf, we see all this green here. That is, what would you call that? Ground tissue, dermal tissue, or vascular tissue?
Ground.
So yeah, ground tissue. And in this diagram, what is the name for ground tissue in a leaf?
Epidermis.
Ground tissue in a leaf is mesophyll. So this line here, the black line around it, what of these three tissue systems is that?
Dermal.
Dermal tissue system. And on this one, it's epidermis. All right, looking at the stem, we've got epidermis, and then all that major kind of beige-looking color back here. That's all ground tissue. But specifically in a stem and root, actually too, there's special names for that ground tissue, which we'll be going over. The ground tissue next to the epidermis, but between the epidermis and the vascular tissue, is called cortex. Ground tissue in middle of all the vascular tissue is called pith. Oh, I forgot to point out the vascular tissue. Vascular tissue here is xylem and phloem of these vascular bundles. And you got vascular bundles in the stem made up of xylem and phloem. And then you've got xylem and phloem in a root with cortex, the ground tissue around, and then epidermis. So, all right, you guys are like, this is a lot of new terms. And honestly, a science class is like you're learning a new language, really. Your brains are gonna be like, oh my gosh, all these new words. And if you've ever learned a new language or you're in a language class, what's like some of the best advice for learning a new language?
Your Latin roots.
What's that?
Your Latin roots.
Latin roots, okay, that's good advice. What else?
Practice.
Practice. How about, is it okay to make mistakes?
No.
No, it has to be perfect. No, I'm just kidding. But practicing is like, when you're trying to learn a new language, you can practice a whole bunch in your mind, but is that going to help you speak it and really get it down? Maybe a little, but really like speaking it. And so in lab today, that's why I'm having you guys try to repeat some of these words we're learning to get familiar with them. And then in lab, say them out loud. Use these new words with each other and start to get comfortable with them. It's okay if you make mistakes. The good thing is you realize you made a mistake and you fix it for the next time. Yes?
Is there a reason in particular around 6 o'clock and 9ish o'clock? There's two phloems instead of a phloem and a xylem for the stem image.
On the stem?
Yeah.
Oh, I see.
Oh, right here.
Yeah.
There's two, there's one.
Yeah, that is weird. That's not normal. So,
Okay.
It's
great
the
observation. It should actually be, usually a vein would have xylem and phloem in it. Yeah. That's
So is that just like a mistake on that part, or is it just?
I'm guessing that's a mistake. I mean, there can be weird examples where maybe, and you guys might see this when you actually take the time to look at vascular tissue under the microscope, which we're gonna be waiting till Tuesday to do that part. That's a complex tissue, and vascular tissue. It's pretty. It's tricky to get down and be able to recognize it. But every once in a while, you'll look at a slide and you're like, I'm seeing xylem, but I don't see phloem here. It should be there, but sometimes there might be some weird part of the plant where you actually, that slice didn't get it. And you might not actually see xylem and phloem in the same area where you should see it.
Gotcha.
So, and I wasn't the artist of that, so for this, so I don't know if that was on purpose or not to show that sometimes you can see some weird stuff like that. But normally you'd have xylem and phloem together. Yeah, good observation and good question. Yeah.
What does xylem and phloem look like under a microscope?
We will get to that, but we're doing kind of like a little bit baby steps. We're jumping through stuff fast, but we'll get to xylem and phloem later. That is what xylem, a part of xylem can sometimes look like. Some of you might have seen a little bit of it when you looked at your banana last time. But we will get into it. Let's work up to xylem and phloem. All right. So, epidermis, periderm, and then with ground tissue, I'm going to say for ground tissue, depending on what plant organ we're talking about, like if we're talking about a leaf, the ground tissue for within a leaf is called mesophyll. I just want to make sure these terms are up here so you can see them. And then for a stem, you guys tell me, looking up here on the board, what do you see there? What is the ground tissue called in a stem?
Cortex.
Cortex. And another one too. Pith. Good job. Sarah, you want to remind them the difference between pith and cortex?
Cortex is just, it's like on the outside of that vascular cambium ring. Oh wait, it's not labeled vascular cambium, but the cortex is on the outside of the vascular cambium
Or
ring.
just say vascular bundle.
Or the outside of the vascular bundle is like that line where the red and blue split. And then pith is kind of just in the middle, and pith is just a bunch of like lame parenchyma cells that look like bubbles.
Not lame, no. They have a function. So I did mention parenchyma is a real common cell type that we're going to see a lot, and it might kind of seem. Actually, Sarah, for fun, draw a big parenchyma cell on the board. This is, I think, where Sarah's coming from with calling them kind of lame. They're not lame. They're cool cells. They do important things. But I am gonna have you in lab like draw a picture of a parenchyma cell you're seeing on your slide, and you're gonna be like, really? Am I supposed to do that? Because you're just gonna go like this. Done. And you're like, wait, did I just draw an air bubble? Because we talked about that, right? Air bubbles look like that too.
Make that together.
But they have important functions. So think about, what is the function, do you think, if most of the cells in a leaf are mesophyll cells, which are actually types of parenchyma cells? What do you think their major function is inside of a leaf? The green part. The green part of a leaf. Photosynthesis. Not lame stuff. It's cool. It's cool. It's like, but yeah, they do seem kind of like, what's the point of that cell? Sometimes you see them and you're like, what? And in the pith, they have a function. They're filling up space and supporting the vascular tissue, but they might not seem like, they're not full of chloroplasts. So you're like, what are they doing? All right. So let's get into trying to think here. Actually, let's continue with the way we're going here with dermal tissue. So let's talk epidermis. So the skin. Epidermis is considered a complex tissue type, tissue system. And it's complex because it's made up of different kinds of cells, different kinds of things are going on there. And so you've got, epidermis is for, I'll just put it here just to help you remember, skin of herbaceous plants, and/or plant parts. Periderm is skin of woody plants or plant parts. And I say plant parts because you can have a woody tree, right? But the leaves are herbaceous and more like, you know, you can bend it, it's not like a woody stem or branch or something like that. So as soon as you're talking like a woody plant or part, that skin is gonna be called periderm. If we're talking like an herbaceous plant, like this celery here, or we have the geranium here, or Zabrina. This is pliable, though some leaves can be pretty thick. But that is what I'm talking about when I say herbaceous. Yes?
Can you plant have holes, or is it just either or?
Yeah, a plant can have both, and that's why I also say plant parts, because you can have a shrub, and it has herbaceous leaves. And even if the leaves are evergreen leaves that are thicker, like a holly kind of plant, or some oak trees are evergreen, like they keep their leaves, they would still be considered herbaceous and would have epidermis on those parts, but then the woody branches and trunk would have periderm. Yes? 2 questions, go ahead. Whichever one wants to jump in first.
Okay, are there any leaves that are considered woody? 'Cause I'm struggling to think of any.
Yeah, I can.
Okay.
Yes?
So is the periderm like bark or something like that, or?
Okay, yeah, good job. Periderm is considered outer bark.
Okay.
Inner bark's something different.
What's that?
We'll get into that later. You want to just know everything all at once. Well, inner bark, I'll tell you right now, has to do with phloem. So that's getting into vascular tissue. But outer bark is periderm. And maybe you guys can, I don't know if my computer's typing all that stuff there. Hopefully that's better. See, a little better. Okay. All right, so epidermis. So let's focus in on the, first off, epidermis. And remember, this is all under the category of dermal tissue. So epidermis, of course, you guys are, I think, we already covered this? Maybe we didn't. Oh, we were talking about cell wall for protection. But same function here for the epidermis and periderm, both are for major function protection. But epidermis can also have other things going on. It also has other things going on too. There's specialized cells that produce wax. And that waxy layer is called the cuticle. So waxy layer is a cuticle. And other substances can be produced too. We also have what are called guard cells, which are kind of these like half donut shaped cells. There's 2 of them together that form what are called stomata. And stomata are the little openings or that allow for gas exchange in the plant. And they're made up of 2 special cells called guard cells. The guard cells guard the openings. And those guard cells, depending on what's going on, whether the plant's hot, if it's hot and dry, those guard cells will release water, or water will leave those cells, and then that hole closes. If they're full of water and turgid, that hole is open, and gas exchange can occur. A major gas that leaves a plant is what? Oxygen. What's another one? Huh? CO2. Normally, CO2 wouldn't be leaving. Normally it's going in. Because plants have a hard time keeping up the concentration of CO2 inside. And so even though they're producing CO2 by cellular respiration, which we'll get into more with mitochondria, they like having a lot of it in there. So usually they would send that out. But another thing.
Hydrogen?
So hydrogen connected to
Oxygen is also
Oxygen.
split out.
So oxygen's leaving. So remember, oxygen gas is two oxygen atoms put together. Water. So hydrogen connected with oxygen forming water, right? So we've got water vapor leaving. That is huge. When a plant's filling up water, you guys, it's pretty amazing that the plant only uses probably like 5% of that water going through the plant. All the rest of it just goes right out. Depending on the plant. If we're talking like cactus in a dry desert, it's not gonna let much of that leave. But if we're talking like a corn plant, that water's going through like crazy. And trees, a lot of water goes up through trees. All right, so that's the guard cells. And they control gas exchange. How much water vapor is getting out, how much oxygen is getting out, how much carbon dioxide is getting in. We have also got trichomes, which are hairs, and then prickles. So here's our. Okay. All right, so here are guard cells. And this is
Those are beans.
and they are green. They do have chloroplast in them. Generally they're green. And then they do have, I'm gonna have just like, you're gonna see there should be a nucleus in them. And then of course, the vacuoles in there as well. All right, and then next to these, this is just a general leaf, okay? We're doing the undersurface of a leaf. Most of the stomata on leaves that kind of grow and kind of go like this, you know? Grasses do this a lot of times. So they've got a different thing going on. But leaves, general leaves that kind of go like so, like we see with this green over here and geranium. There's definitely an undersurface and an upper surface, right, to that leaf. That undersurface is where your guard cells are going to be, where the stomata are. Because if you think about how these leaves are arranged, their upper surface is kind of facing what? Aiming towards the sun. And if that opening, the stomata, a lot of whether or not it's opened or closed is whether or not how much water vapor is leaving and how much it wants to conserve that water. They're going to be on the bottom to be in the shade and help conserve water. So you're not going to see, you'll hardly ever see any stomata on the upper surface. So when you guys are looking at, you're going to do a special epidermal peel in lab today. You're going to do 2 of them. And
It's really tricky.
it's tricky. It's really cool too, because you see some cool stuff. Especially like, it might take you a couple tries to get a good one. That's okay. It's tricky because I want you to try to get like 1 cell thick peel, but that's the tricky part. You know that you're getting down into the mesophyll if you see a lot of green on your peel. Because these epidermal cells might be a little more clear-ish. They're there for protection, they're not really there for photosynthesizing. So you're gonna have more of like a clear looking peel for the geranium. And classic, not always, but classic general epidermal cells kind of look like puzzle pieces. So you've got these other cells around. Those would be like general epidermal cells. And then you might have a cell over here. I think I drew one of these the other day, where this one's kind of got an interesting shape, and it kind of keeps going and going, and maybe look like that. What do you guys think that is? It's a trichome, I know. So the trichome, it has a base that looks like the other epidermal cells, but then the cell continues. And this might actually be a separate, 3 separate cells. and then you would see nuclei in each of those, if that was the case. You may or may not see a trichome that has a gland on the end that might look like that. Or you might actually see a cell. What is going on out there? What's going on there, guys?
They're listening to heavy
Replacing
metal.
the handles on the doors, and they were doing it. They've been following me all day.
Yeah?
I was in the rock room, and they were doing it on the doors on the outside there. And then I was in lab in 1401, and they were doing it on the doors on the next there. And now they're here.
Why are they following you?
I don't know.
Alright, so a glandular, and when you notice, when you touch these leaves, there's a little bit of stickiness to them, and you can smell that geranium oil. And that is being produced by glands. So your hairs might not look, I'm not specifically drawing geranium here, okay, just so you know. Your drawings are gonna look different. This is just giving you an idea of different components of what an epidermis might look like. But epidermal cells are different with different species. And there are some really crazy hairs out there that might look like, almost like a tree. And then there's the base, there's the cell. And each one of these might be a separate cell within the nucleus. And maybe that is another cell. Okay? So trichomes can look really different depending on what they're for. Yes?
I'm assuming this isn't the scale, but what magnification would we have to be to see something like that?
You can see them probably. You would be able to see them at the lowest magnification of 40.
Like pretty clearly, or?
But you're gonna want to zoom in at least to like 100 to 400. Yeah. 400, you might actually, depending on how big the hair is, might be coming off your field of view actually. So they're pretty good size. And as you've noticed, I'll be bringing in some different things to pass around. But sometimes it's like almost furry. Some leaves have so many large hairs that one of them is, you guys know mullein? Another common name is toilet paper plant. Charbon plant. No, I'm just kidding. They have their leaves are very soft.
Yeah.
And furry.
What do
I
they
was
do?
trying to point that plant out to you actually.
What's that? Oh, so this is showing some leaves have a very thick waxy cuticle. Some not as thick, but they all have some sort of thin waxy cuticle layer. And here you can see this one shows the stomata. Single stomata, and let me write this out here.
You got the same slide I
Stomata being plural, stoma being singular. Okay. We'll get into this in more detail later. This just shows an image of what I talked about how the guard cells function in controlling the opening, whether the opening is open or closed. And we'll get into that in more detail later. Okay. So here we can see, oh great, you guys will hear me say, oh, another term, there's lots of terms. Sometimes, this is different than cholenchyma, sometimes mesophyll cells are called chlorenchyma cells. Sorry, I don't have you learn that for exam purposes, but you may see it. So these are all mesophyll cells, or according to this, chlorenchyma cells, but they're a type of parenchyma, and they have vascular bundles. Here's the epidermal layer, upper with a waxy cuticle, lower waxy cuticle, and what's this? Yeah, so those are the two guard cells. So here's showing a cross-section through a leaf that has very branching trichomes. And a proper term for if you're talking about all the trichomes of a plant, you'd say the plant's pubescence. And that's proper botanical term. And here you have another showing another type of branching trichome, and so forth. All right, let's see, what else did I want to mention here? Oh, forgot to say prickles. If you've got a rose, so here's a rose stem, and normally people will call these structures on here thorn, but that is not proper botanical terminology. These are considered prickles because they're actually extensions of the epidermis. So if we're talking specialized cells or components of epidermis, prickles are that. So roses have prickles, not thorns. So, you know the song, "Every rose has its thorn." Who sings that? You guys don't know who sings that?
You apparently.
Is it? No, not me. Is it Poison?
Poison from Street Fighter?
I think it is.
I think it's Poison. Yeah.
Verified by the internet.
Yep. All right, cool. All right.
Who's this Poison?
It's, wouldn't the song sound better, right? If they did it botanically correct.
Not really, no.
Every rose has its prickle. Just like every cowboy sings a sad song or something like that. No, it's supposed to be prickle, prickle. A thorn is another botanical thing. Thorn is a modified branch or stem, not epidermis. Okay, so these are prickles. Anyway, can anybody think of any other plants that have prickles other than roses? Yes.
Raspberries.
Awesome. Good example. So, and periodically I'll bring in samples and you'll get to see those up close when we talk more about either that group or whatnot. Or we're comparing prickles to thorns to spines is another botanical term of another pokey thing that's not. They're all three very different things botanically. Yes?
So prickles are just like a overgrowth?
Mm-hmm. Of that epidermis, right. So it's kind of a compilation or a whole bunch of cells coming together and then they form a point right there on the epidermis. All right. How are we feeling about epidermis?
Pretty hungry.
All right. So that's epidermis. Now periderm is skin of woody plants. It's the outer bark. We'll get into this. We'll get into this part a bit later, and we'll get into it in a lot more detail when we cover woody stems. That's when we get into it. All right. So let's now get into ground tissue. Okay, so ground tissue, remember in leaves it's called mesophyll. In stems it's the cortex and pith, depending on how that vascular tissue is arranged in the stem. And, oh, shoot, forgot to say, and roots. All right, so what's happening here with ground tissue? Ground tissue, we've got parenchyma cells, parenchyma cells, and sclerenchyma cells. And mostly ground tissue, it's like the support, right? It's what's surrounding the vascular tissue. It's supporting the vascular tissue. It provides like substance to that. Imagine a leaf without ground tissue, right? It would just be epidermis and vascular tissue and veins, and it's got to have that substance, right? So it's supporting. It also provides for storage, depending on what those cells are and what they're doing. So parenchyma tissue / cells, guess what parenchyma tissue is made of? Parenchyma cells. So that's why I say tissue/cells. And remember that lovely drawing Sarah did on the board of a parenchyma cell? Now you can see they just look like bubbles. Their cell walls are not very thick. And in a leaf, they are the mesophyll and they're conducting photosynthesis. In a potato, that's what you saw. They're conducting what? All those cells you saw in the potato slice.
Storing nutrients.
They're storing. They're storing. And sometimes there's secretion involved. If you take a pine needle, for instance, and we slice your pine needle, look in there, you're going to see special resin canals. Guess what's lining a resin canal? Parenchyma cells. Parenchyma cells are lining resin canals, and they are producing resin and secreting it into those resin canals. So parenchyma cells, they do a lot of different things. And okay, we flesh of fruits.
Wait, Suze.
Yeah.
They're not boring because they are able to differentiate into any cell type that they want. They're like the plant version of stem cells.
Awesome. So can you say that again? Louder.
Parenchyma cells are actually really cool and they're super not lame at all because they are basically like undifferentiated, and so they can become whatever type of plant cell, like whatever other type of plant cell is throughout the entire plant. They can become like vascular tissue, epidermis, like they can, that's why when you take a piece of leaf, a succulent, like that succulent leaf is filled with parenchyma cells and it can grow a whole new plant.
Sounds boring.
Stem cell. Yeah, it's like stem cells.
So it's right here.
No idea.
Most of these cells, they retain their meristematic ability. So what Sarah just described is called meristematic ability. So if you take that apart here, stem, and you think of stem cells, not the stem like a plant organ stem or a branch, but stematic in that that cell can become any kind of cell. And so it retains that. So that is really a really cool feature of these kinds of cells. And another important thing is they are alive at maturity. Remember those cells that you saw that TEM of the persimmon seed, those hard bony cells that have really thick cell walls, nothing in the middle? Those ones are dead at maturity. They have a function of just structure. They don't need to keep their living part, going. But these guys do. They stay alive at maturity. All right, anything else I want to say about parenchyma at this point? Okay, so we talked about bryophytes, mosses, liverworts, hornworts, not having vascular tissue. They don't have vascular tissue, but they have epidermis, and they have a lot of parenchyma cells. They've got ground tissue. And a good portion of their ground tissue is different kinds of parenchyma cells. All right, the next one is parenchyma. Parenchyma tissues are made up of parenchyma cells as well. And these are really interesting. If you can kind of make sense of this, this is showing how they might look. Two different examples, mostly like this. And a parenchyma cell has uneven cell walls. So, whereas parenchyma, those are really thin cell wall, and it's like the same width around the whole cell. Okay, parenchyma. So this is like showing the parenchyma cell. Parenchyma cells will have thicker areas and thinner areas and thicker areas and thinner areas and thicker areas. So uneven cell walls. And these cell walls, the thicker parts are pretty thick, so you're going to be looking for these you're going to make your own microscope slide of celery. So celery, what do you guys think you're eating when you eat celery? What plant organ?
Stem.
It seems like stem, and it is stem-like. But if we're talking about what a full leaf is, a leaf encompasses, okay, here's a stem, here's a leaf. The leaf encompasses this whole part, the leaf petiole and the leaf blade. And when you're eating celery, you're eating the leaf petiole. You're eating this part. Except celery leaves, so the celery stem is this short thing down here, and then it has all these leaves that come up. And I'm running out of space here. It has all these leaves. And the more the blade part's up at the top, but that's a petiole. And so we take this. Who likes to eat celery in here?
I can
I
use
like
that.
it. I like how crunchy it is, and it has an interesting flavor.
Raise my hand
It'd
because
be
I
an
was kind of
A-tier
scared
plant,
she'd
except
throw
it's way too
some.
stringy.
Doo-doo-doo, here's our beautiful celery.
Something Dr. farted.
I know the string, get the string stuck in your teeth. Yeah.
That brings it down to a C tier.
That's good for you.
And those stringy parts are kind of stretchy. A lot of that material, yes, there's vascular bundles in there, so there's veins in here, and you can actually see those ribs. Inside those ribs, there's going to be vascular tissue in there. But a lot of what makes this so like nice and supportive, like stays nice and solid, right, is parenchyma. Parenchyma cells. Yes, there's parenchyma in there too. Yes, there's vascular tissue in there too. Yes, other things. But a big component is, other than parenchyma, is parenchyma. And you're going to do a slice through this and try your best to include some of the epidermis with the ribs, because that's where you're going to really see the parenchyma cells. And when you get those strings, a lot of that component is parenchyma, and they're stretchy. And so a big part of a function for parenchyma is structure for parts of the plant that are herbaceous and still growing. So another special kind of cell that gives support to plant parts that aren't growing anymore is called a fiber cell. And generally we see those in stems, not leaves so much. You might sometimes have some fibers in leaves too, but parenchyma fiber cells generally are dead at maturity. Parenchyma alive at maturity. They can like stretch and have more flexibility than a fiber cell in a way. So how many people like having peanut butter with their celery? All right, I did bring some peanut butter. So you're gonna be making slides of celery, but I'll have some cut up and you can smear some peanut butter on them and have that as a snack. So I do try to bring snacks in here and there. So you are what you eat. No, what you're looking at, you're eating, and hopefully it like all sinks in, and you're like, I'm eating these delicious parenchyma cells and parenchyma cells, and I'm never gonna forget those terms and what they do because they're now a part of me, right? That's what I'm hoping is gonna happen. Yes.
I got you a question. So are the stringy bits like the fiber cells you were talking about?
Parenchyma, these guys.
Okay.
Mixed with some, there's most likely some xylem and phloem in there too. And I'm gonna have you try to find xylem and phloem in there. Not today's lab, but next lab, I'm gonna have you try to find some xylem and phloem in celery as well. Today, the focus is gonna be on parenchyma and parenchyma. So you do get so stems and leaf petioles. So herbaceous stems, leaf petioles, around veins like those stringy things in celery. Those are helping these cells around here are going to help to give protection and support to the vascular tissue and the whole structure as well. All right, primary cell walls. Remember we talked about primary versus secondary cell walls? Primary cell walls, parenchyma and parenchyma cells, that's all they have. Whereas when we get into these other ones, sclerenchyma, fiber cells, they develop secondary cell wall and are even thicker. Fibers and sclerids. All right, so, I forgot to mention, I don't have it written down here, but write down in your notes, parenchyma tissue and cells remain alive at maturity, just like parentheses, alive at maturity. And that actually reminds me when I'm talking about notes. There is, I was gonna see if anybody would be interested in this classroom, even though I do have lecture slides as a PDF online, right? You guys have access to that. If anybody feels like, if you're like, I'm like awesome at writing notes, taking good notes, and feeling like you feel good about that, and would like to make some extra lunch money, I'd love to have a note taker in this class. So if you want to think over that and let me know at break or after after school, then let me know, and I'll send you over to. I'll tell you where to go. It's over in it's Pace, and it is over in the cafeteria area. But I'll give you more specifics. I have to look at the room number. But yeah, so come and let me know, just so I know if we get that covered in here. All right. I just remembered that when I was talking about notes. Yes.
It's $1.50 per class day, right?
No, it's more. I think it's like $3 to $4. I'll double check on that too. I'll get the room number and I'll get the proper amount, but I think it's $3. Has anybody been a note taker before? Do you remember how much?
It was about $3 to $4.
Yeah, I think it's $3 or $4, which you do that a couple times a week, and adds to one lunch. I was like,
You'll be
it
short
adds to
$2
something.
actually because of lunch.
It depends on where you're eating, where you go. You just buy lettuce and celery. You need more than that, just joking. All right, any questions so far?
Maybe some rice.
On what we've covered. I love discussion happening, but let's have it so the whole class can hear what's going on. You might have a good question or a comment? No? You might have a question or comment? Want to share? All right. So, we've talked about a lot so far. Let's talk about sclerenchyma and then have a break. And then I'll have you get your microscopes and then we'll jump into what's happening for lab. All right, so sclerenchyma. Everybody, remember, let's practice this. Everybody together. Sclerenchyma. Sclerenchyma. We should do like a rap. Right? That would be really cool.
The horrors.
Sclerenchyma, and
Dude,
then
I'm
you
gonna
can
be
go in
going
other
this
words
class.
that rhyme with
I think it's like
Emma.
hallucinations from hunger.
All right, so sclerenchyma offers support similarly to cholenchyma, except what's the big difference? No longer growing, right? So cholenchyma offer support and have these uneven cell walls, but their primary cell walls are still stretchy to parts that are still growing, so they can grow with that organ. Whereas areas where there's no more growth happening, fiber cells can be produced, which is one of the examples. And they have really thick cell walls. Secondary cell wall, primary plus secondary cell walls. And they come from the term hard, because they are hard. And that secondary cell wall has a component in it called lignin, which is even more dense and offers more structure than cellulose. There's cellulose there too, but the lignin definitely helps with that. And most of these are dead at maturity, though this example here on the side, that still looks alive, doesn't it? Still looks like there's stuff going on inside those cells. All right. Fibers, sclerids are examples. Okay, I'll go back to that in just a second. Let me show you fiber cells first. So fiber cells, if we look at a cross section of a fiber cell, it looks like a donut with a little tiny space in the middle where the living cell once was. But it's mature. When it's mature, it is dead, and that middle part is not, there's no more functioning nucleus. There's no more cell membrane in there. It's just cell wall. Yes?
Is the lignin present in both the primary and secondary cell walls?
It depends on the species. Some species produce more of it, but my understanding is you would find it more in secondary cell walls. So you wouldn't really find it in like the parenchyma stuff, in walls of cell walls, primary cell walls of parenchyma cells. They don't need that. Their function is not for like strength and support like we see it in these other kinds of cells. Yes?
So is parenchyma a fiber also, or no? Okay, it's just structure.
No, and good point here. Let me add to, we're talking about sclerenchyma, and one of the examples of cells under that umbrella category of sclerenchyma cells, sclerenchyma tissue, are fiber cells, which are different than parenchyma. And remember, parenchyma and parenchyma, those are parenchyma cells and parenchyma tissue. Parenchyma cells and tissue. Sclerenchyma cells and tissue. But specific types of sclerenchyma cells are fiber cells and, and sclerids.
Can we look at the fiber cell slide?
Oh yeah, we're gonna go back. I just wanted to point out the two kinds. Yes, we're going back. We're gonna go back. Don't worry. All right. So, okay. Wait. Look here. Fiber cells, sclerids. Okay. Now, tell me. What do you think this is? So, two types of sclerenchyma: fibers and sclerids. What do you think this is? Anybody want to guess? It's hard, it's really hard to tell what's happening here.
Wait, what are we doing?
I'm asking, okay, I'm just kidding, okay, everybody look. These are fiber cells.
Yeah.
Whoa, fiber cells, sclerids.
Can we look at
So what is this? Very good, yeah. These are sclerids, for sure.
Can we
So-
go back to the square slide?
We're gonna look at all the slides, okay, guys? I just wanted to see if you could guess what these were by looking at those other slides really quick. So the two major types are fibers and sclerids. This is a sclerid. And what do you guys think? Let me pull this other thing up really quick. Where is our? I think it's right here. Going back here. This is a little tricky, but what do you guys think this is? That form like, you guys looked at this already, this is like the cells that produce the pits, peach pits. They are sclerids, but, so if you imagine a lot of sclerids together can form bony material. Because those cell walls are so thick. And normally they're dead at maturity. But what you see here is that these ones are still alive at maturity, and honestly, the ones you're gonna see in lab are gonna probably still look kind of alive as well. Fiber cells are dead at maturity. And let me show you side view, difference between fiber cells and sclerids. So if you look at a cross section of a fiber cell, looks like that. Looks like a wheel or a donut, but longitudinally, so long ways, depending on the species, they're going to be a little bit slightly wider in the middle and tapered at both ends. So if you were to slice right through this middle part, that's what you see, okay? So fiber cells, long and thin, tapered at both ends, really thick cell walls, impregnated with lignin, primary and secondary cell walls there. And so primary cell wall and a secondary cell wall as well. Now sclerids might not be as perfectly round. They might have, they look very similar in cross section, but might not be as nice and round. But then longitudinally, they look something like that. So if we're cutting right through that, since the size is not exact, I was kind of running out of space here. But they're kind of like little blobs, little short, wide blobs, and hard, like hard little bony blobs. Does that make sense? Whereas fiber cells, long, thin, also hard, but long and thin. And hemp fibers are fiber cells. And you're like, but those are pliable, but they're thin, but they're really strong, right? They're really strong. And you would have to really pull on those to get them to break. And here, this actually is an image of a slide like you guys have already seen. Remember when you were looking for crystals in the tilia slide? This is actually from that. And areas where you were kind of seeing some crystals were kind of maybe around here, or maybe it was up in here in this area. But what I'm wanting you to focus in on here, which you didn't really notice probably maybe back last time when you were looking at this slide, there's a lot of fiber cells in there. And they just look like these sort of squished strawberry donuts. And they're dead at maturity. Looking at these cells, what do you think these cells are? Thin walled, kind of look like bubbles. They're parenchyma cells. Very good. And some of these parenchyma cells actually have material in them, kind of showing that when this slide was made, they were alive. They were alive at maturity. All right. So sclerids, shells of nuts and stone fruits, like we talked about, peach pits, cherry pits. That hard bony material are just a whole bunch of sclerids all clumped in tight together. And in lab, we're going to have you actually find sclerids by looking at pears. Like the flesh of a pear is mostly parenchyma cells. When you're eating, like I said, when you eat like that fleshy fruit, you're mostly eating parenchyma cells that have a lot of sugar in them. You're also going to notice like a grittiness. Anybody like to eat pears? You know what I'm talking about? The gritty. There's like a gritty texture. And it's interesting. Pears are closely related to peaches. They're actually in the same plant family, or major group. They are in Rosaceae, which might seem kind of crazy. They're in the rose family. And there's a lot of things in the rose family that have those hard pits. Pears don't have a hard pit, but somewhere in that evolution, those sclerids are there. They're just not forming a pit. They're just kind of spread out in the flesh. Yes?
Aren't strawberries also in the rose family?
Yeah. I know. Rose, like some plant families, like the bean family, the legume family, everything in that plant family has the same kind of fruit. A legume. That's the kind of fruit that they have. Other plant families, they just have one type of fruit. The rose family is really unique in that it has so many different kinds of fruits in there. And so you have to look at other features to help you realize, wait, that's part of the rose family. The way the flowers look, if you compare strawberry flower to a raspberry flower to a cherry flower to an apple flower, they all look really similar. Yes?
To be classified as a fruit, does it have to be like grown above ground, or can it also be underground?
That's not a fruit. A fruit classification is that it is ovary tissue that has seeds in it, or at one time had seeds in it. We might have like selected for seedless, but that bears seeds, or should be bearing seeds. And there are some weird examples where sometimes the fruits can develop underground, like peanuts is an example of that, where the fruits actually end up going down and into the ground. Hence, another common name for peanut is groundnut.
I've never heard that.
You hadn't heard that? Anybody else heard that before? Groundnuts, peanuts? No?
Ground bean.
Ground pea?
Ground bean.
Ground bean? Maybe, yeah, I think in Africa. So, another question. No, okay. All right. Sometimes sclerids can have really crazy shapes like this one. This one's found in water lily leaves. All right, I think we're good. Maybe? Yes? Oh, let's look at this. Ooh, no, wait, one last picture. Well, we already saw this, okay, but let me point out that, please. Quiet, guys. All right, remind me what these lines are.
The vascular?
No,
Middle
connective
lamella.
tissue.
Okay, this is middle lamella.
That.
It's like a plasma.
Very good, keep going, keep going.
Plasmodia.
Yes, good job, you guys. See, I love it.
Oh, I see.
Plasmodesmata. You guys put it all together. Plasmodesmata. Right here, it's calling it a pit canal.
Plasmodesmata.
That is another term for plasmodesmata. It's a canal that connects two cells, and if you look on one side, it looks like a pit.
That's not, no idea.
That's why it's sometimes called a
I
canal.
thought you were gonna say.
But it's plasmodesmata. And you can see that it's connecting these together. This is what I want you to look for when you look at the, I almost said peach, pear. When you look at the pear flesh, you're gonna make a microscope slide of that. Get inside that pear and get a little bit, squish it on your microscope slide, a couple drops of water, cover slip on top, and what generally happens, like you see here, they're kind of together. Over here are parenchyma cells, and there's a group of sclerids right here that you're gonna do something where you get a piece of paper towel so you don't get your fingerprints, or the cover slip paper. You know that cover slip paper is kind of a pain, it kind of flies around, you're getting your cover slip. You're like, where's that cover slip? Those dang papers. Those papers do come in handy. You can actually use them for the wicking. Remember how you wick the iodine over? You can use them for that. You can use them to make something, piece them together and have a big thing of tissue paper. No, just kidding. But you can also put them on your cover slip and set your slide down and put your thumb on there and squish. And maybe even sometimes twist a little. And otherwise you just have this big clump of sclerids and it's just gonna be this dark mass. And you're like, what is that? And you don't wanna get your fingerprint on the cover slip because that makes it hard to see. So the paper just keeps your finger juice from the cover slip. And you squish it to help separate out those sclerids. But not too hard, you don't wanna break the microscope slide as well. All right, final slide, and then we'll go for a break. Sclerid, remember I said it's kind of this kind of short, kind of blob, hard blob. That's what it looks like from the side view. Ooh, I forgot to draw the difference between parenchyma and parenchyma. Parenchyma cells are kind of like a little bit stretched out, slightly. Like stretched out, but not as much as fiber cells. Fiber cells really stretched out and elongated. And it's crazy to think they all started as one kind of cell and then differentiated depending on where they're located into whatever their job's going to be. Certain genes get turned off, turned on, and then they start becoming whatever kind of cell they need to be. Yes?
What would happen to parenchyma cells when that part of the plant stops growing? Or do they only occur in parts of plants that never stop growing?
Good question. And sometimes you'll see this with parenchyma cells as well. If they don't have a function where they will die, but then they just kind of get either, have you guys, well, one thing you look at on Tuesday is squash stems. And if you've ever looked at like a squash stem, it's hollow inside, where the pith kind of, at one time it was all parenchyma cells, and maybe there was some parenchyma there too, but then they kind of degrade and kind of break apart. They kind of get taken, the place gets taken with something else. And, but parenchyma cells do hold that potential meristematic activity where they can become something else if they don't die out, and depending on if they're needed. Oh, but this is good. This shows these bigger bubbly cells here, thin-walled cells here, and then the unevenly thickened cells here. The cell wall is like white and sort of shiny. That's what you guys are gonna see with the celery. You're gonna look for something like this. What do you think these bigger, thin-walled cells are?
Parenchyma.
Yeah, so you've got parenchyma cells coming right up next to parenchyma cells. And then right here, these are the epidermal cells here. So this is like a rib of celery. Epidermal cells, parenchyma cells, and then parenchyma cells right here. And I'm gonna ask you to find a spot where you see the difference between parenchyma and parenchyma. And then fibers. All right. Let's go ahead and break, and then when you come back, actually, I'm gonna make anybody who can remember to come up here and grab your lab. Like I said, we're doing part of it today, part of it on Tuesday. Also have stuff left out for the last lab if you want to revisit green banana, some other stuff. So when you come back, grab one of these, grab your microscope, and we'll be ready to go at
3.
Yeah. Try to be here just a little bit before 3. So we're ready to get started right at 3.